Hello class, this is Dr. Anderson and we will be talking about the male reproduction system. So as usual, we start with our learning objectives. We are going to talk about the role of the Y chromosome in maleness. So XY is the genotype and maleness is the phenotype. We're also going to talk about the structures that support the formation of sperm and the transport of sperm. This is not an anatomy class, but it can be helpful to talk about the structures as we're talking about the function of things. There are some very important support cells called Sertoli cells found in the testes, and they surround the gametes as they're forming, and they have a regulation function in spermatogenesis, which is the process of making sperm. Males are straight negative feedback kind of people, and so we are going to talk about the negative feedback regulation of testosterone secretion and of spermatogenesis. These processes start at puberty and reach their full function at maturity, and then males are potentially capable of producing testosterone and sperm for the rest of their life. And we're going to talk about situations where there's too much testosterone or not enough, and how that interacts with the regulation of the reproductive axis. And then, of course, we want to talk about the functions of testosterone. So around week 7 in the embryologic development of a male, some things start to happen to get that male to the phenotype of maleness. And as I've said previously, sex is genetically determined. XX is the genotype for females. XY is the genotype for males. And all people start out androgynous. They start out with 2 sets of undifferentiated tubules, and then very early in embryonic development, the Y chromosome, which contains a very important gene called the SRY gene, that gene is transcribed and the protein products are made, and that stimulates development of the testes and allows for the production of testosterone. Testosterone is then able to promote the differentiation of one set of those tubes into male structures, happens to be the Wolferian tubes. And then there are Mullerian tubes. Those are actually female tubes. And the expression of the SRY gene also allows the production of Mullerian inhibiting substance or Mullerian inhibiting hormone, and that causes the female tubules to go away. So early in development, the SRY gene is expressed, and the male embryo starts to produce testosterone, which brings about male structures and produces Mullerian inhibiting substance, which makes the female structures go away. So when we think about reproductive physiology, we talk about a primary reproductive organ. The primary reproductive organ is the testes, and the testes has 2 jobs: to make gametes, which in the male are called sperm, and to make sex hormone, which in human males is called testosterone. The testes are suspended outside the abdominal cavity in adult males, and there's a countercurrent mechanism that has to do with the blood supply supplying the testes that allow the testes to be a little bit cooler than abdominal temperature. So here, this is not from your textbook, but this is what we want to concentrate on right here. So this is the vas deferens, and there's an artery in the vein. And warm arterial blood is going towards the testes. Cooler testicular blood is going in the vein towards the body. And what will happen is heat from the artery will directly transfer to the vein, and then that heat is carried back towards the body. And oh my goodness gracious, I've just realized I have a typo here. The transfer of heat from the artery to the vein. I have that backwards in my PowerPoint. I will fix that before class. But warm blood from the artery then goes straight to the vein, and then the heat is carried back to the body. Now, why is that a good thing? It means the testes is, say, 3 or 4 degrees cooler than body temperature. And that just so happens is an optimal temperature for good development of sperm. When sperm are too warm, quite frankly, if the testes are too warm, and that can be because of tight clothing or things like that, that can interfere with sperm production. So here's the picture from your textbook showing the testes with all its tubules. Let's get my laser pointer, Lucille, back here. So these are the seminiferous tubules. Lots and lots of tubules, lots of surface area for producing gametes. Those collect in some tubules, and those tubules eventually collect into one tubule called the vas deferens. The vas deferens then takes the sperm into the abdominal cavity where it can join with semen and prepare for the ejaculation process. About right here is where the ligation process for a vasectomy would take place. So, contraception is a complicated process. Male reproduction or male contraceptives, for a long time the major option was using condoms. There are issues with condoms. You can have a surgery, vasectomy, but the way that surgery is performed by many surgeons, it's an irreversible process. Giving some kind of pill that would inhibit testosterone production is a problem because that interferes with the sex drive. So what's an option for contraception in the male? Well, one option that I don't talk about here is trying to interfere with the receptor-ligand interaction between sperm and eggs. The one method that I highlight here, and I have a link if you'd like to follow that if you're interested, is the idea of reversible inhibition of sperm under guidance. And basically what happens is a polymer is injected into the vas deferens, and then it hardens. And so then you have a physical blockage of the vas deferens. The sperm cannot be added to the semen, and therefore the ejaculate doesn't have sperm in it, and the male has contraception. And then in some formulations, there's a substance that a man can drink, and then when it leaves in the urine, causes the what do I want to say? Dissolves the block so that the block dissolves and leaves in the urine, and then the passageway is preserved, and then the man could be fertile again. So this is a interesting new thing on the horizon. All right. So the seminiferous tubules are where the sperm are made. And so there are lots of tubules, and in the lumen is where the sperm are going to arise. And here's a nice blow-up of that. We have something called the blood-sperm barrier. Then these very complicated shaped cells surrounding the gametes, those are the Sertoli cells, and they protect the sperm from outside influences. Then here are the stem cells for the gametes, and they develop, and as they go towards the lumen, they get more and more mature. Outside of the seminiferous tubules are some cells called Leydig cells. Those are the cells making testosterone. So you see a couple of things. FSH, follicle stimulating hormone. Now, FSH was named for female functions. Then endocrinologists figured out males have the same darn hormone. So FSH, follicle stimulating hormone, stimulates the Sertoli cells and promotes spermatogenesis. And then LH, luteinizing hormone, stimulates the interstitial cells of Leydig to produce testosterone. Testosterone has effects throughout the body, but testosterone is also required for spermatogenesis. So here's some of the anatomical features of the male. Here's the testes suspended outside in the scrotum. There's the epididymis, where many of the sperm are stored. There's the vas deferens entering the abdominal cavity, looping around the back of the bladder. Now it becomes the ejaculatory duct, and secretions are added. Secretions of the seminal vesicles, secretions of the prostate gland, and secretions of the bulbourethral glands, which are also called Cowper's glands. And then semen leaves through the erect penis during intercourse or during ejaculation. Now notice that males have this long tube called the urethra, and it's serving two purposes. Sometimes the urethra is carrying urine, and other times the urethra is carrying semen with sperm. But there's a valve system here so that those two fluids aren't traveling at the same time. So seminiferous tubules is where the sperm start, stored in the epididymis, vas deferens, ejaculatory duct, urethra, and penis. There's a cute little mnemonic here, 7-up, SEVE, N stands for nothing, UP. And then seminal secretions, the seminal vesicles, the prostate gland, and the bulbourethral gland together are going to add the fluid to the gametes, so that fluid is called semen. So semen is the fluid for sperm transport. It comes from those seminal vesicles, prostate glands, and bulbourethral glands, which would all be examples of exocrine glands. Semen has some useful substances. One of the things semen has is bicarbonate, because what we're going to learn is that the vaginal ecosystem is very acidic. There's a population of bacteria that produce acid that protects the female. However, then the males needed to evolve an adaptation to withstand vaginal acid, and so semen is very rich in bicarbonate. It's got some energy substrates because sperm have to be able to make ATP so the sperm can swim. And then there are other factors that we would call motility factors. Those encourage the sperm to swim. Energy substrates, there's some fructose and there's some citrate. So those are carbon substrates that the sperm can burn in the mitochondria to make ATP. All right, so what happens in spermatogenesis? So I'm assuming most of you have had biology and have studied the process of mitosis, which is one DNA replication followed by one division, so that you have two identical cells produced from that process. Meiosis is one DNA replication, some genetic assortment, followed by two divisions, so that four cells that are all genetically unique are produced. So in males, this process starts with cells called spermatogonia. Spermatogonia are stem cells. Stem cells divide mitotically to maintain the population of those stem cells. Some of the stem cells then progress into meiosis, whereas other stem cells stay undifferentiated and continue to divide mitotically. So males potentially can produce sperm the rest of their life because they maintain a population of those spermatogonia. So cells differentiate and enter meiosis. Those cells are now called primary spermatocytes. and they have 46 replicated chromosomes. During the first meiotic division, the cells or the chromosomes line up on the metaphase plate. They line up in tetrads. Homologous chromosomes line up in tetrads. They exchange some DNA in a process called crossing over. And then we have the first meiotic division. So homologous chromosomes are separated. The importance of that is that heterozygous traits are separated. So if your mom has straight hair and your dad has curly hair, and you have a curly hair gene and a straight hair gene, you have to separate those traits. So you can make straight-haired gametes or curly-haired gametes, but the gametes cannot have both alleles. So notice we go from 46 to 23 chromosomes. Those chromosomes are duplicated, but you have separated those homologous chromosomes. Then without a replication, the cells divide again. That's the second meiotic division. And now we have 23 single chromosomes in a spermatid. But this cell cannot swim. So the cell has to differentiate into a spermatozoa, also called a sperm, so that the DNA can travel. Those sperms are basically DNA delivery cells. And sperm with vaginal intercourse and reproduction, sperm basically have to swim the equivalent of the English Channel. So they are specialized for that. So in the head of the sperm is the nucleus. There's a covering called the acrosome that releases enzymes that allows the nucleus to make its way through the cells around the oocyte so that fusion can happen. In the sort of mid-piece of the mitochondria of the sperm are all kinds of mitochondria. So the mitochondria are taking the substrates in the semen, breaking that down and forming ATP, because ATP is required for the movement of the flagellum so that the sperm can swim. So with sperm production, it takes approximately 64 days to make a sperm. And typical males make about 30 million sperm per day. So males are about quantity. That's their biological strategy. So males are continuing to make sperm. Those sperm are protected by the Sertoli cells. The Sertoli cells also make something called androgen binding protein. Androgen binding protein helps keep testicular testosterone high. And how does that work? Well, testosterone is lipid soluble. It can go wherever it wants. But if you have a binding protein, some of that binding protein can hang on to the testosterone and keep it in the testicles. Why is that important? Because testosterone is required for spermatogenesis. So again, all stages of spermatogenesis are found in the Sertoli cells. The gametes developing, I'm sorry, I think I should have said all stages of spermatogenesis are found in the seminiferous tubules. Those gametes are surrounded by Sertoli cells. That's what I meant to say. And so we have stem cells that are undergoing mitotic division. Some of those progress into meiosis, so you have a primary spermatocyte that divides into two secondary spermatocytes, divides again into four spermatids, which then develop further into sperm. Sertoli cells, in addition to making androgen-binding protein, also make inhibin and activin. These are very important peptide hormones. They regulate a hormone from the anterior pituitary called FSH, but they do a lot of reproductive functions, and we don't begin to understand what they are. Quite frankly, if you take knockout mice and prevent them from making inhibins and activins, the mice die. So you have to do studies in heterozygote knockouts, and I'm waxing too long on this, but this is a hard thing to study. But scientists continue to plug away at trying to figure out what these inhibins and activins do. All right, so here's a feedback mechanism for hormone production. Remember, guys are straight negative feedback all the time. So the hypothalamus secretes a releasing hormone called gonadotropin releasing hormone. Gonadotropin releasing hormone goes through the portal system, binds to receptors in the anterior pituitary. The anterior pituitary have cells called gonadotrophs, and they produce two hormones. One is FSH, follicle stimulating hormone, and the other is LH, luteinizing hormone. Now what's weird is that these hormones were first discovered in females and were given female names. And then the scientists went to the males and said, whoops, they have the same darn hormone, but we're just going to keep the female names. So sorry guys, we have female names for your hormones. So FSH binds to receptors in the Sertoli cells. Now, the Sertoli cells produce some growth factors that stimulate spermatogenesis. And when spermatogenesis is at an adequate level, the Sertoli cells make inhibin. And inhibin then inhibits the anterior pituitary. Making sperm is very energy intensive. So males don't want to spend extra energy making more sperm than they need. So there's a negative feedback mechanism that makes sure sperm production is not too high. It won't enhance fertility, and it wastes a lot of energy. So you want spermatogenesis to be just right. Now there's another negative feedback mechanism going on here. So the anterior pituitary makes LH, luteinizing hormone, that binds to receptors in the Leydig cells, and the Leydig cells produce testosterone. Some of the testosterone is required in the testes because testosterone is needed to make sperm too. Now, what do you call this relationship? Is testosterone permissive for FSH? Is FSH permissive for testosterone? Nobody agrees on that, but I would say that the sort of the most favorable way to describe it is that FSH and testosterone are synergistic in working on the Sertoli cells to produce sperm. If that does not make sense, we're going to talk about it in class again, and you'll have one more chance to go over this concept. Testosterone then goes into the blood, binds to sex hormone binding globulin, circulates throughout the body. It's responsible for the sex drive. It's responsible for the development of the reproductive tract, for the maintenance of muscle mass, of all those sorts of things. When testosterone levels are getting too high, they feed back and inhibit LH in the anterior pituitary and inhibit GnRH in the hypothalamus. So there's some specific inhibitions here. So I want you to think about this is a negative feedback mechanism for regulating testosterone, and then this is a negative feedback mechanism for regulating spermatogenesis. So consider a man who has one testicle surgically removed. And why could that be? It could be injury, or it could be testicular cancer, so one testicle is removed. So let's think about the short-term and the long-term effects on testosterone production. Well, initially, what's going to happen if one testicle is removed? Testosterone production is going to decrease. You removed one of the testicles. So there'll be less testosterone feeding back to the hypothalamus and the anterior pituitary. That should allow the increase in GnRH, increases in FSH and LH, and FSH and LH will work on the remaining testicle to promote normalized function. So actually there are men that have one testicle that are fertile and able to perform sexually, and the system is able to compensate. I also want you to think about a man with a depressed pituitary function and low testosterone. We talked in class the other day about how someone could have a tumor in the anterior pituitary that produces growth hormone. Let's say somebody has a tumor in the pituitary, but what the tumor does is compress and basically squish, compress the cells that make FSH and LH. So there's low pituitary function. As a result, there's low testosterone. Now, have you seen the commercials about, oh, you're not getting older, it's low T, so you can take a testosterone supplement? Well, could we give this guy testosterone and normalize his sperm production? Well, we could give him testosterone and normalize his testosterone levels, but to get sperm production, what else does he need? He needs FSH, and FSH production is being compromised in the pituitary. So you'd have to either give both testosterone or FSH, or a good thing would be what's going on in the pituitary and could we address the primary issue? Now, what is testosterone doing? Well, it's essential for spermatogenesis. It works with FSH to produce sperm. It's important for feedback regulation to the hypothalamus and the anterior pituitary. It develops male organs in the fetus and in puberty. It's responsible for secondary male characteristics, so a larger larynx and a lower speaking voice, a larger muscle mass. Males tend to be taller and bigger. Higher levels of secondary, having a beard is facial hair is a male characteristic. So things that, allow men to look like men. Testosterone is important for sex drive. However, females make some testosterone too, and it's responsible for sex drive in females too. That's kind of interesting. Growth and sealing of the epiphyseal plate. Remember that from our discussions of growth hormone. It starts out as cartilage, but once that's sealed, and there are actually lots of hormones involved in this, but testosterone speeds up that process. And males tend to have a higher hematocrit, or red blood cell count, than females because testosterone is a powerful stimulant of erythropoietin, which is a cytokine that promotes red blood cell production. So what happens with puberty? Well, when we are children and we produce a small amount of sex hormone, that sex hormone is enough to inhibit the hypothalamus. And if we inhibit the hypothalamus, we inhibit the anterior pituitary. But the thing is, around age 8, the brain seems to notice the lack of sex hormone. The brain matures, and the brain sort of loses its sensitivity. It used to be very sensitive. Little tiny bit of hormone would turn it off. Now it's less sensitive. That tiny amount of hormone is no longer inhibiting the hypothalamus. So the hypothalamus starts to produce GnRH, then LH, and testosterone comes from the testes. And the male organs start to grow. So the first sign of puberty in a boy is a growth of the testicles, growth of the penis, appearance of pubic hair, appearance of axillary hair. Those things are part of the things going on with puberty. Testosterone accelerates growth, sealing of the epiphyseal plate, and males reach their adult height. Now, females have a growth spurt too, but their growth spurt happens earlier. I don't know if you remember, think way back in 4th or 5th grade when the girls were taller than the boys. The girls have their growth spurt earlier, so then their epiphyseal plate seals sooner. Boys have their growth spurt later, so they get to grow taller before they seal off their plate. It's just kind of... Now, there are exceptions to this, but this, sort of in general. Alrighty, so let's think. There's a man who's taking androgen. He's a football player. He's a baseball player. He's, I don't know what's going on. He needs, maybe he's in an occupation where it's intensive physical labor and he needs to be strong. So he says, I'm going to take an androgen. And that androgen steroid, I'm going to take that because testosterone and androgens cause the buildup of muscle mass. I need to bulk up so I can be strong, either to be an athlete or to work in a particularly strenuous field. Well, what would happen in terms of feedback? Well, let's think about this. You're adding an androgen. That androgen maybe could be converted to testosterone or mimics testosterone. High testosterone is going to feed back to the hypothalamus and do what? It's going to inhibit GnRH. High androgen is going to feed back to the anterior pituitary and inhibit LH. So if you took a blood sample from this guy, he would have high testosterone, but that testosterone would not be coming from his testes. It would be coming from the testosterone that he is taking. And this can be a problem. All righty. So that is the end of our presentation. And think about your questions. Keep thinking about physiology, and I'll see you in class. Bye for now.